Earlier last week, scientists from China reported that they've created the first pig-monkey hybrids, or chimeras as they're known. Chimera is a Greek word used to describe a mythological creature that had a lion's head, a goat's body, and a serpent's tail. The animals created in the lab in China don't look like the chimeras described in the mythological creature, but they do resemble the overall nature, which is animals created from two different species that were combined. In this case, the scientists used pig and monkey cells to create a pig-monkey chimera, essentially a pig that contained some of its cells that were coming from the monkey. So why were these creatures produced in the lab? Well, the overall goal is to eventually one day create pig-human chimeras that could be used to grow human organs such as the lung, heart, or the kidneys and be used for transplantation. Although currently there are no ethical laws that allow for such creatures to be generated, this step in being able to create pig-monkey chimeras would one day open up the doors to create specific organs that could be used to harvest for transplantation. So how did the researchers go about in making these pig-monkey chimeras? Well, the first thing that they did was take the ovum or the egg from a female pig and fertilized it using the sperm from a male pig donor. In addition, they isolated embryonic stem cells from a monkey. Then they labeled these embryonic stem cells with a green protein, which allowed them to be tracked during the developmental stages. And also, once in the pig, they could identify these green cells that were coming from the monkey itself. They then waited for the fertilized egg to go through multiple rounds of division until it reached the blastocyst stage. In the blastocyst stage, the inner cell mass is where the embryonic origins of the animal will come from. Within this inner cell mass, they then injected the green labeled embryonic stem cells from the monkey, essentially creating a blastocyst that contained cells both from the pig and the monkey. They then waited and injected these blastocysts into female pigs and waited for them to give offspring. After the offspring was born, they analyzed these pigs to see what proportion of them contained green cells, essentially coming from the monkey itself. The researchers then isolated the various organs from these animals, such as the liver, lung, spleen, skin, and uterus, and analyzed them to see what proportion of these different organs contained green cells, which meant these cells came from the monkey. And what they were able to observe is that certain organs had way more of these green cells than the other ones. As you might be able to tell in these pictures, these green cells that are lighting up are coming from the monkey itself. So what they demonstrated was that they are able to create these pig-monkey chimeras, and moreover, different organs are able to continue to maintain these cells that are coming from the monkey itself. Now, this is exciting, but the frequency in which they were able to obtain these chimeras was very, very low. In fact, the researchers only obtained 2 pigs out of the 80-something different pigs that were born. Moreover, the 2 that was born as a chimera did not survive more than a week. Now, there might be different reasons for why the survival was really poor. One of the suggestions is that in general, when they perform in vitro fertilization using different species as donors and recipients, the animals don't survive for too long. This has been long known in the field of in vitro fertilization biology. The scientists in China are continuing to work out different methodologies to improve the efficiency and the survival of these animals. Although this was the first demonstration of the ability for scientists to create animals that survive beyond the embryonic stage, this was by no means the first time these kind of chimeras have been created. Two prior studies in the past have generated similar chimeras. One earlier study was able to demonstrate the ability to form rat pancreas inside a mouse. A later study by the Belmonte group was in fact able to create human-pig chimeras. For ethical reasons, these human-pig chimeras were not allowed to survive beyond embryonic stage, for the fear that some of the brain tissue of the pig might actually come from the human cells. Therefore, it would be unethical to let these animals be born and then sacrifice them. As a result, these studies were stopped at the stage of the embryonic development. But recently, Belmonte's group has suggested that they have created a human-monkey chimera in collaboration with researchers in China. Although the ethical manifestations of such experiments are still hotly debated, the technical and scientific challenges that these kinds of studies are able to overcome will definitely change the way transplant medicine will be performed in the next 10-15 years. Moreover, it opens up basic questions about development and our understanding of cell biology.